zinc oxide, and other nanoparticles materials have demonstrated the strong antimicrobial properties. And we all know that due to the extremely small size and large surface area, so these particles can interact effectively with the microbial cell membrane microbial and can disrupt the cellular function, and even they can damage the microbial DNA. So because of the multi-targeted mechanism, now the nanomaterials or nanoparticles become the much more means can say the better approach for the antimicrobial as an antimicrobial agent. So at the same time, we and also the another advantage is the targeted drug delivery. Nano carriers can help the delivery the drugs or the antibiotic directly to the site of the infection. So this may improve the therapeutic efficiency, and it may reduce the toxicity and the other side effects. So with this brief introduction, I would like to invite all the respected participants to actively share their ideas, their knowledge, their experiences, or the research insight so that we can have a meaningful and productive discussion. Once again, thank you all for the joining today's session, and I warmly request everyone to participate actively in this discussion. So now I request all the participants, if you have any research experience, insights, or perspective, you can share with the participants. So Dr. Prakash Salami sir has joined. Good evening, sir.
Good evening. Good evening.
So we may begin our discussion with the introduction. So just unmute yourself and introduce, and then we can share your research experience or your ideas based on the nanotechnological strategy for antimicrobial. So here the Is Das Ganga, whatever the name is showing in my list, Alok Rai, Arpita Panda, Asima, Asmita Chakrabarti, Ayush Jasrotia, Deepika Kar. Please unmute and introduce yourself first. Hello. Am I
Hello.
audible? Or?
Yes,
Yes,
yes.
Alok Rai. Yes.
Good evening, sir.
Good
Myself,
evening.
Dr. Alok Rai. I'm working as a professor in Department of Microbiology at St. Kisimal Porval College, Kamti. So basically, from last 10 years, I'm working on nanotechnology and antimicrobial some drugs, basically some loaded nanoparticle drugs.
Yes, sir. Then
So
today we are very fortunate that today we have joined with us.
yeah, thank you, sir.
Yes, sir. Please, sir. Hello. Yes, right, sir. Yeah.
Sir, I have introduced myself. Later, after introduction of all the participants, we can discuss, sir.
No, sir. You can share your research experience, sir.
Okay, okay.
Sure, sir.
Uh, Sir, basically, I'm now working on some consortium-based nanoparticle from algal cyanobacterium, or even I have worked on Rhizoctonia solani fungi-based nanoparticle, which was not been done by most of the researchers in India. Now, I am also working on some synthesis by actinomycetes also. So, sir, we
Mm-hmm.
have now synthesized quantum dots, size less than 10 nanometers, mostly 6.5 to 7 nanometers now. And we
Mm-hmm.
have achieved it by consortium of two cyanobacterium. So this nanoparticle has more efficiently, it works on antimicrobial. It shows very good antimicrobial, and it also shows very good antifungal activities. And some of nanoparticle, we have tested it on DNA damage also directly, and we have found some nanoparticles. They are very good in damaging the DNA. And I have also tried some antioxidant activity and anti-diabetic activity in lab by comparing it with metformin as a drug.
Mm-hmm.
And so I am getting very nice results these days, and I'm happy that I'm working on biological synthesis. So it is more better than chemical synthesis. And So now my next paper is coming in scientific report, and we are publishing
Okay.
it on basically we have synthesized nanodots, which have better biological properties. So I can share that we can do lots of new research in this area, basically in nanotechnology. And now I have also tried to load some drugs on this nanofibers, and it also enhance the activity of that particular drug. So these
Mhm.
things also have been done with help of some pharmacy collaborators. And in this way, we are working on different aspects of nanotechnology.
Okay.
So I have shared my
Medicine.
ideas, sir, and if anybody have interest
Yes,
in that,
sir.
yeah,
This one is our objective, sir.
okay, okay.
Discussion so that we can help the other researchers.
Mhm. So we can discuss some new ideas. Basically, now, sir, most of the people were
Namaskar,
Mhm.
sir.
synthesizing nanoparticles.
Alex,
Yeah.
sir. Alex,
Yes,
sir.
ma'am. Yeah, ma'am.
Myself, Dr. Ganga.
Yeah.
Sir, I'm working on the content dot side. So you said you are working on the content dots from the microbial side, right?
Yes, yes, yes.
Sir, which color pattern you are getting, blue or green or red? Which one is getting for you from micro?
No, no, we are working on cyanobacteria, ma'am. It's a green in color, and it reduces most of the salts which are using, like
No,
silver.
sir. My question is, you're getting the fluorescence, right? Which color of I mean, the fluorescent is which color it is? What you are getting?
Fluorescence means what type of what you are asking? I don't understand. And
CD, content dot. You said you're working on content dot,
no,
right?
actually, zeta potential we have done it, madam, and we have seen some. We are not working on that. We have only seen the size of this quantum dots, and
You're not
we
seeing
are
the fluorescent part of that.
fluorescence part. We are not working on that, madam, because that is basically not in our lab, and we have just go for the zeta potential of that particle only, and
Okay.
size basically and properties we have seen. That's all.
Are you getting the uniform size, sir?
No, madam, not uniform size.
Uh,
Not uniform
that's
size.
what the problem. When it comes to the industry level, they're asking for the
Mhm.
uniform size. So we have to have a discussion. Even I am not getting the uniform size. For that, for that sake, I asked you.
No,
But for
you
the
know,
chemical, that is
yes.
another drawback in that case. So
Mhm.
yeah. And
But
I think,
but
can you share your number, sir? We can have a collaboration if it is required.
yes, yes, ma'am. Okay, I will share with you. No
Mine,
problem.
yeah. Thank you, sir.
Actually, at what area you are working, madam?
Sir, I am working on the plant-based CD road, sir. Basically, I am also microbiology background, but my
Mhm.
students are working on that aspects, sir.
Okay,
So
okay.
I think we can have a collaboration each other in that.
Yes, yes. Actually, basically, we are looking for this AMR also, antimicro resistance bacteria. We have tried this synthesized nanoparticle, or you can say some quantum dots. They are giving more better results as compared to because
But
of the
sir,
surface.
but when I was my doubt when I have tried with antimicrobial activity, it is not working. So
Okay.
that's what I wanted to ask you in person, like how this can be. I mean, yeah,
Yeah,
I can be
we
improved.
are regularly doing antimicrobial activity in our lab,
No,
madam.
I just wanted. Yeah, I will. I have my synthesized one, so I can even send to you.
Okay, okay. What type of media you are using? Basically, you are dissolving in which we are using DMSO as solvent for this.
Have you cross-checked the DMSO as a standard also?
Yes, yes, standard also. But we are not
Maybe
getting.
that is a reason, sir. We are dissolving it in normal this one, so not in DMSO. Maybe that is one reason. Anyway,
Okay,
we can
DM.
have a discussion, sir. If you can give your number.
Yeah, yeah. I will give my number. No, no problem, madam. Just
Okay.
okay. I
And
will
also,
share it.
sir, right, sir, you can share your Gmail means mail ID in our chat box so that anyone can
Okay,
contact you.
okay. That I can
Okay,
do, sir. That I can do, sir.
okay,
That I can.
okay.
Thank you.
Thank you.
Thank you, sir.
Any participants, if you have any question to Ganga madam or the Rai sir, then we may ask now. Or you can put your question in chat box. I can read out this. Hello.
Which kind of pathogens also he is testing? That can be actually interesting information.
Yes, sir.
I actually was trying to means log in that time, so I couldn't hear some of this stuff. But I think his research experience looks very interesting up today's this roundtable discussion. So I think he can actually elaborate. We should give him some more time to tell about his research activities. What do you say,
Yes, sir.
Shivariji?
Yes, sir. Sure, sir. Alok, sir.
Yes, sir.
Yes, sir. We request you to, sir, elaborate more so that you and Ganga madam is also here.
Sir, basically, I am focusing on, you can say, biological material basically, and I am trying to mix some bacteria or try to make synthesize some nanoparticles because most of the organisms are not been explored. My two papers in, was published in very good journals now, and it was from we have synthesized it from Rhizoctonia solani, or you can say it was my first report. We have synthesized my then I have also used some microalgal like Tetradermis obliquus, which is a very common algae found in fields, and we have reduced some of the nano materials, and like that we are doing. We are trying to find out some. We are exploring some new material for the reduction of these salts. So I'm not using any chemical methods now, and being a biologist or being a microbiologist, I'm trying to make most of the nanoparticles by bacteria or fungi, or you can say actinomycetes or other types of bacteria. So and
So your purpose of biosynthesizing nanoparticle is only for antimicrobial activity,
no,
or
I'm also trying it for antimicrobial, antioxidants, anti-diabetic, or
wonderful.
even anti-cancer also we have done. And we
Yeah,
have
very
found
good.
very good results in case of lung cancer or ovarian
You are
cancer.
from which institute, you told?
I'm working in SK Porwal College, Kamti, Nagpur.
Dr. Alok, you have come from which institute?
I am from SK Porwal College, Kamti. It's in Nagpur, Maharashtra.
Alok, can you hear me?
Yeah. Please. आवाज आ रही है सर मेरी आपको? हेलो.
हाँ सर, आ
No,
रही है
little
सर.
bit network issue is there.
ओके नो, यहाँ तो नेटवर्क इश्यू तो नहीं
नहीं
है
सर,
सर अभी तो
आलोक सर
फिर.
की आवाज क्लियर है सर.
नहीं,
हाँ.
मेरे तरफ ही network issue है. No, you come from which institute? I was asking.
I'm working as a professor in Department of Microbiology, sir. SK Porwal College, Kamti, Nagpur.
Oh, very good.
And you can
So this is not
basically
PG center, right?
we have PhD center. Yeah.
This comes under Nagpur University.
Yeah, it's a in Nagpur University. Yes.
Yeah, very good.
So one of my publication is just coming in scientific report, and I can share with you all of you. With we have
Sure sir.
synthesized some smalls. You can say nanoparticles. We have given a name like small silver nanoparticles. Small means it is less than 10. So we are having some doubt at that time that it is a quantum dot or not. So now we have tried to synthesize it in more precise way by changing some temperature or pH of the reaction. So
So
by
antimicrobial
this,
activity you are checking in vitro, right? In a test tube,
yes,
in
in
a
vitro. I
petri
have
plate only.
all the yeah, in vitro well diffusion method I am using, and almost all organisms I have a range of candida, or you can say
Uh-huh.
AMR bacteria. I have just Klebsiella or
Oh,
other
very
type of
good.
bacteria we have. So the application part is very important, sir, because I was working with some chemistry persons before this biological synthesis, but They are unable to publish most of the paper because they don't have good applications. Now we have started applying it in biological field. So we are getting very nice paper. And I have also published one paper in ACS journal, and it was been on a some cell line related to insect cell line, and we have seen most of the activities in that. Yeah. So lots of things can be done. It's a very interesting subject. So if anybody interested, please contact me, sir, or I can help, or if I need your help, I
Yes.
can just with you all.
Sir
No
has
problem.
shared his number and also the mail ID. So you can
Yeah.
copy from there. So Dr. Ganga madam, now I request you to just share your research experience here, ma'am. Hello. Dr. Ganga madam. Are you there, madam? Now the Dr. Seema Tiwari, Dr.
I'm here,
Rupa.
ma'am. I'm here, sir. I'm here, sir.
Yes, yes.
Yeah, myself, actually, I'm working on the same way as in condensed dot, not in the microbial manner, but I am from the phytochemical manner. Basically, I'm a microbiologist. But what I'm seeing is when I'm taking from the phytochemical sample, we are getting different fluorescent manner, like green fluorescence as well as blue fluorescence, and even in the green fluorescence. And my one of the area is like um, a dye preparation, which I'm because a patent part is going on. And another one is I made a biopurifier, but that is already got patented from algae. And another one is we are working on the polymer aspects also. So, but I have some I stuck it in the content dot part. So that's what I asked Alok sir help. And much more thing is like for me getting publishing is very difficult because I have developed a PGPR consortium for plant promotion, and it has got a very good activity for I mean curcumin generation in the in turmeric, and the curcumin level is very high when the consortium is applied. But somehow I don't know, my paper are not getting published. So many people advise me if you have some collaboration with some people which has already been, because I'm new to this area, newcomer. So if you have a collaboration with somebody which has already been published many papers, it would be better. So I need that collaboration, that part as well, sir.
Okay. Thank you, madam. Anyone, if you have any
Dr.
question,
Tiwari can help you for publishing a paper in good journals.
Okay, sir. I need a number also now, sir.
yes, madam. So Dr. Prasoon sir has joined now. Dr. Bandana. So you can unmute and just introduce yourself. Dr. Rupa, Dr. Seema Tiwari, Kumar. Yes, please.
Hello, sir.
Yes, madam.
Sir, this is Dr. Bandana. Sir, I'm a senior resident in BJ Government Medical College, Pune. I am here for learning purpose, sir.
Okay, thank you, madam, for joining. Dr. Rupa, Dr. Seema Tiwari.
But Dr. Bandana can tell about her activities.
About her. Hello.
Hello, sir.
Yes, madam.
Sir, currently I'm posted in bacteriology section only, and
Yes.
I have not had much
So you
exposure
will
to
come across with the
nanotechnology as of
AMR,
now.
antimicrobial resistance. You will see
Yes,
the
yes, sir.
antibiogram test, right?
Yes,
These
sir.
are very common.
Yes, sir.
MIC determination. You know.
Yes,
So
sir.
these are very
Those things
important
we are
in
doing, sir.
microbiology also. In a medical
Yes, sir.
microbiology.
Yes, sir.
Dr. Abhijit Patil.
Abhijit Patil.
Good evening, sir. This is Dr. Rupa here.
Yes, madam. You're audible, madam.
Yes, sir. Sir, actually, I am also here. Yeah. I'm also here for learning. Like I have worked on antimicrobial resistant bacteria. I isolated E. coli from fish sample and its environment. Just wanted to explore what all are the options. So I'm here for the learning. वायफाय कनेक्टेड आहे नको कनेक्ट करू किंवा मी इथे लॅग व्हायला लागलाय
Hi, good evening, all. I have a question. Can I ask?
Mhm. Sure.
Yes. Actually, sir, I am working with Pseudomonas putida for degradation of pesticide. That pesticide is chlorantraniliprole, which is from anthranilic diamide group. Sir, actually, I have made zinc oxide nanoparticles from Pseudomonas putida, and now I am a bit confused with antibacterial and antifungal activity of that. Actually, sir, it is not showing zone. So against E. coli and against there are 5 pathogens we have taken, but it is not showing zone, sir. So what should I do, or matlab, how should I conclude that? Is it good if zinc oxide doesn't show that doesn't kill no, these organisms?
No, madam, actually, it should kill the organism. Zinc is very good, showing antimicrobial activity. How you are trying, I don't know. Madam, please dissolve in DMSO and go for sonication of the sample. Because dispersion is very important in nanotechnology. If the particle is dispersed, it will have more good activity. If you don't use the sonication activity or sonicator, that will not do the well. So you can try
Okay.
it. You can try by dissolving it in DMSO.
Actually, sir, I have a very, matlab, I don't know whether it's a name question or what it is, but sir, these organisms are found in soil, right, sir? So
Okay.
if I am killing pesticide and if this oxide, zinc nanoparticles are also killing the soil microflora, okay,
Mm-hmm.
so how it is good, sir? Because see, soil microflora ke andar the microorganisms are also getting killed by these nanoparticles, and
Okay.
हाँ
Yeah.
nanoparticle जो है वो pesticide को भी मार रहा है, which is a good thing, but वो organisms को भी मार रहा है, तो how it is good, sir? मुझे वो मतलब I don't know
No, that
कि
is not good.
name
That is not
question है
good.
शायद
It
से.
depends upon which bacteria you are killing. If it is a good bacteria, then it is not a good thing. If it is killing the flora of the soil.
हाँ
So,
तो सर Pseudomonas aeruginosa, this E. coli, they all are microflora of soil ना. It is present in soil. But अगर हम ऐसे नैनो पार्टिकल्स बनाएं और जैसे कि अभी बहुत कुछ चल भी रहा है कि नैनो
so E.
पार्टिकल्स
coli
से
can be,
तुमको
if you kill, madam, if you kill E. coli, that will best. क्योंकि E. coli तो flora होके भी कोई फायदा नहीं है. That was not been used by any have any good activity for the plants. But Pseudomonas or you can say Bacillus subtilis or other bacteria, they show very good PGPR activity in case of you can say I mean, in case of growth promotion. So it is very important that how you know that this degrade the pesticide. The nanoparticle has degraded the pesticide. How you have concluded it?
हाँ हाँ सर UV I did UV and
No, you
उसके
have to go for
अंदर
GCMS, madam. You have to go for GCMS. And
हाँ हाँ
first you have to take the pure form of your pesticide and you have to go for the what are the products, what are the means degraded products of this.
हाँ
Uh,
that's what sir when
One
I
of my
am doing
student has
UV.
done it. Now he's degraded the pesticide by cyanobacteria, and we have found out the secondary metabolites, means the metabolite which is not dangerous for environment, and we have found it in case of GCMS, and we are now going to publish it. So you have to
Okay.
first take the pure form of the pesticide. That is very important, madam.
Sir, we have said this chlorantraniliprole pure pesticide. I have bought it from West Bengal. Now, sir,
Yes, sir.
when I was doing UV also, so what happened? 30 minutes के लिए we have kept it in dark. फिर उसके बाद में 10 मिनट्स के लिए we
Yeah,
have
the
we
reading
took,
was reducing. That you are
yes,
saying कि वो कम हो रहा है धीरे है ना?
yes, it
ये
sir,
तो
it
that is
reduced
a good.
till 20 minutes. 20 minutes the peak was
Okay.
coming down.
But
Okay.
what
So
are the
30
हाँ
minutes में 30 minutes में it went sir up. Then 40 minutes में it went up. So I think so कि कहीं ना कहीं that male principal compound is getting breakdown into byproducts.
yeah. Actually, that can be seen only by GCMS or LCMS. We can predict you can say see the structure. Basically, what are the byproducts it was being produced by the breaking by this enzymes. Means, आपका तो ये है ना क्या नाम है आपका तो नैनो पार्टिकल है तो वो क्या ब्रेक कर रहा है कौन सा ग्रुप को तोड़ रहा है वहां पे वो देखने के लिए आपको जीसीएमएस करना ही पड़ेगा मैडम देन इट विल बी
ओके ओके
क्लियर
ठीक है तो वो सर 30 मिनट्स जहां से पीक ऊपर जा रहा है वहां से आई डू जीसीएमएस या
नहीं आप उसको डायरेक्ट जो आप कर रहे हो ना उसको दैट सैंपल यू सेंड फॉर द जीसीएमएस दे विल गिव द रिपोर्ट कि वो क्या बन रहा है मतलब व्हाट आर द बाय प्रोडक्ट्स ऑफ दैट डिग्रेडेड पेस्टिसाइड
मतलब यूवी से ट्रीट करके देंगे या यूवी के बिना ट्रीट करके दें सर
तो नहीं आप यूवी के जैसा आप कर रहे हो वैसे कीजिए ना यूवी से ट्रीट करके दीजिए मैडम बिल्कुल तो आप देख रहे हो ना कि रिड्यूस हो रहा है तो उसी स्टेप में दे दीजिए ना उसको जीसीएमएस को
ओके 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 डन
यस
ठीक
यस
है
यस
सर आप आपके कौन से स्टूडेंट ने किस टॉपिक पे ऐसा कुड यू प्लीज शेयर द सम डिटेल्स सो दैट आई कैन रेफर सर और साइट भी हो जाएगा पेपर
मैडम मैं भेज दूंगा आपको उसमें मतलब ये मेल पे पूरा डिटेल नहीं बता सकता बट ही हैज यूज्ड साइनोबैक्टीरिया बेसिकली दैट वाज नॉट बीन यूज्ड बाय एनीबॉडी साइनोबैक्टीरिया तो मैंने उसको बताया था कि साइनोबैक्टीरिया के साथ ही हैज ग्रोन दिस पेस्टिसाइड एंड वो थोड़े दिन बाद हमने देखा कि उसमें ये जो पेस्टिसाइड है वो रिड्यूस हो रहा है कम हो रहा है इवन द ग्रुप इज चेंजिंग दैट मीन्स हाँ रिएक्टिव्स जो हैं वो काफी ये हो रहे हैं मेटाबोलाइट्स जो हैं वो दे आर नॉट डेंजरस फॉर ह्यूमंस तो इस तरह से मैं थोड़ा शॉर्ट में ही बता पाऊंगा अभी बिकॉज
यस
इट्स
यस ओके
इसमें
गॉट इट
पाथवेज बताने में थोड़ा टाइम लगता है मैडम उसको
करेक्ट हाँ सर हाँ
हम्म
एक्चुअली और सर मुझे एक और चीज पूछनी थी द जिंक ऑक्साइड नैनो पार्टिकल्स जो मेरे हैं सर
हम्म,
उनका जो लैम्डा मैक्स और ये पेस्टिसाइड का
हाँ
जो लैम्डा मैक्स है प्योर का सर वो
हाँ
240 से लेके 260 के बीच में ही आ रहा है मतलब ये
ओके
पीक भी सर ज्यादा ब्रॉड नहीं आ रहा है ऑल्सो लाइक बहुत ही कंजेस्टेड है पीक तो उसके लिए क्या
हम्म।
कर सकते हैं बिकॉज दिस पेस्टिसाइड इज नॉट कलर वाला तो ये कलरलेस है पेस्टिसाइड सर
नहीं
जब
तो
इसको मैं ना
कॉन्जुगेट्स मैडम यू हैव टू यूज सम कॉन्जुगेट्स जहाँ पे आप उसको कॉन्जुगेट करके यू हैव टू मेक ए कलर रिएक्शंस ऐसा भी कर सकते हैं आप फॉर दैट यू हैव टू कांटेक्ट सम केमिस्ट्री पीपल जो आपको पता है कि रिएक्शन के साथ मतलब कलर दे कैन पुट सम एजेंट तो उसके साथ वो रिएक्ट करके कलर देगा तो वी हैव आल्सो डिजाइन सम कंजुगेट्स की भाई ये कलर क्योंकि कलर नहीं रहेगा तो फिर वो बहुत दिक्कत होती है उसमें यू नो रेंज का विजिबल
वो
रेंज में
वही
नहीं
वही
आता मैडम
वही सर क्योंकि ना मेरे जिंक ऑक्साइड नैनो पार्टिकल भी उसी रेंज में दिखा रहे हैं 240 टू 260
नहीं
लैम्डा
तो
मैक्स आ रहा है और इनका भी वही आ रहा है तो अब
सो
ये
यू
नहीं
कैन
समझ
यूज
में आ रहा
फॉर
है
सम
कि
कलर डाई और कॉन्जुगेट्स बनाना पड़ेगा वो आप कर सकते हो मैडम वो कोई भी सम केमिस्ट्री पर्सन का हेल्प कि वो कैसे करना है उसको एंड वो फिर
ओके
कलर आने का तो वो रेंज में आने से यू कैन विजुअलाइज इजीली या
थैंक यू सो मच सर फॉर
थैंक यू
गाइडेंस
Sir, I have one more
मैडम
doubt. Suppose we have identified two pathogens from rice, new ones. For that, we have
Hello.
tried with some of our the organism, endophyte organism, which is having the antimicrobial activity. We didn't find none of them having antimicrobial activity. But when we take nanoparticle out of this organisms, that nanoparticle is showing some activity. So without that organism is not showing any antimicrobial activity, but their zinc nanoparticle which is produced from that is showing the activity. So will that kind of organism can be taken into account?
Ma'am, you are asking to me or somebody else?
In the
Hello.
forum, sir, I'm asking. Anybody can ask.
Ma'am, can you repeat the question? Actually, थोड़ा sound ये हो रहा था मेरे को.
Sir, we have 10 microorganism, which is 10-15 microorganism, which is from isolated from as an endophyte, plant endophyte.
Okay, okay.
So none of them is showing antimicrobial activity against. We got new two pathogen, two fungal pathogen and one microbial pathogen from our turmeric samples
Okay,
or
okay.
ginger leaves. Okay, it's called ginger root or turmeric root. So none of them is showing any antimicrobial activity. But when we prepare the nanoparticle out of those some organisms, that nanoparticle is showing antimicrobial activity. So somebody told me like since the organism is not showing the antimicrobial activity, that single nanoparticle which is produced out of the organism cannot be taken into account. But is it true or not?
No, no, ma'am. These two things are different. You see, when you're growing or when you're seeing directly applying this bacteria for antimicrobial, it is very difficult because I have one bacteria, actinomycetes, which shows very good antifungal activity. So, what we are doing is that we are giving such type of conditions which can help them to produce those metabolites which are having some antimicrobial or antifungal activity. So you have how you have performed. I don't know कि आपने कैसे किया उसको ना how you have put the organism directly to
No,
the.
sir, it is not showing antimicrobial activity, that's for sure. But when we prepared nanoparticle out of
No,
the extract of
understood how you have tested that this organism is not showing any antimicrobial activity.
that
How you have.
MHA plate agar assay, that well diffusion assay.
No, ma'am, that will not work. Actually, you have to synthesize or you have to grow this bacteria in some conditions or sometime it will take 2 to 3 days to produce some metabolites, special metabolites. And then that metabolite can be spin out from this media and that can be tested for antimicrobial. वो करके देख सकते हो आप अगर उसमें नहीं आ रहा तो भी कोई प्रॉब्लम नहीं है because you see in nanotechnology what happened the metal gets reduced, so if the size is very small the metal is always toxic to any bacteria, so it will 100% shows antimicrobial activity most of the synthesized nanoparticles because of the size, because of this toxicity. So we can't compare these things, man. This both things are different. I think so.
So you mean to say that I can consider that it's nanoparticle for my antimicrobial work, right?
Yes,
Okay.
100%. You can consider.
And
Yeah.
ma'am, sir, regarding the spinning part, whether I should mix that with any amyl alcohol, acetyl alcohol, or alcohol, or is
No, no, no,
directly you can
no.
take the supernatural?
Directly you take the metabolites, which was extracellular metabolites, or you can try that if it is showing. Sometimes it shows antimicrobial activity.
Okay, sir. Okay, sir.
Yeah.
Thank you. Thank you.
Thank you.
So one question is from Disha Chauhan. Does anyone know about the acute bacterial conjunctivitis? This question in the chat box. Dr. Prabal Chauhan, Dr. Sunitha. Yes. One from the Kunjal Jinjade. We had synthesized selenium from plant extracts but we are not getting the lambda max so in our research. And one from the how to confirm the nanoparticles are synthesized. Hello.
Hello, sir.
Yes, Arif, you may ask your question.
Yes, sir. I'm a research student from Shiva University, Kolhapur.
Sure. Yes.
माइक्रोबायोलॉजी सब्जेक्ट मेरा जो टॉपिक है वो नैनो पार्टिकल से रिलेटेड है.
Mhm.
तो मैं अभी फिलहाल में नैनो पार्टिकल सिंथेसिस कर रहा हूँ. तो मेरा क्वेश्चन था कि कैसे कंफर्म किया जा सकता है.
Nanoparticle synthesis हुआ ही है ना?
Yes, sir.
The nanoparticles are synthesized. This you want to confirm.
सर एक्चुअली जिंक ऑक्साइड नैनो पार्टिकल्स मेरा सब्जेक्ट है. तो जिंक एसिटेट मैंने यूज किया और वो जैसे पेपर में बताया वैसे प्रेसिपिटेट मिल गया. वो सेंट्रीफ्यूज कर दिया. तो पहले जो मेरा क्वेश्चन है कि वो नैनो पार्टिकल ने कंफर्म करने के लिए कौन से टेस्ट करना पड़ेगा.
Okay.
First, you have to go for XRD.
XRD.
Yeah, you have to go for what crystalline size it has, and second is FTI. You can see whether which salt you are reducing is there or not. That is also very important.
Yes, sir.
These two things are very important.
Yes, sir. Please repeat.
You can use first of all XRD
ओके सर, हाँ
to see the crystalline size. If it is very nearby nano particle, then you can say कि बना है करके. And
अच्छा,
second thing, you can use FTIR.
FTI, ओके
हाँ, simple. These two thing will give you a picture कि ये बना है करके. Yeah.
अच्छा सर, और एक क्वेश्चन मेरे को था कि मैंने अभी XRD किया है, लेकिन वो
हाँ.
XRD के सैंपल जो है उसकी रीडिंग आ गए, उसका मतलब कैसे कंक्लूड कर सकते हैं वो रिजल्ट जो है, वो उसका एनालिसिस कैसे कर सकते हैं हम?
मुझे भेज दो, मैं कर दूंगा, उसमें क्या है.
सर थोड़ा मेरे को थोड़ा यही द बेसिक डाउट था सर, इसके लिए मैंने
नहीं, we have some software also and we will do it. और अगर आपको सीखना है तो you can come to Nagpur or sometime we will do it, no problem.
ओके
और
सर,
अगर बेसिक आपको हाँ, अगर बेसिक अभी करके चाहिए होगा तो करके दे देंगे एनालिसिस, उसमें भी नहीं, बिकॉज वी हैव सम बेसिक फैसिलिटीज वी हैव इन आवर
चलेगा
लैब.
सर। मेरे पास XRD की रिजल्ट आ गई, लेकिन उसके आगे कैसे क्या करना है वो नहीं समझ में आ रहा सर, इसके लिए
यू
सर
सेंड
आपका।
इट, आई विल गिव कंक्लूजन और
ओके
क्या क्या पिक्स मिले हैं और कैसे उसको उसका थीटा वैल्यू क्या आई है और वो आपका बना है कि नहीं, आई विल टेल यू, नो प्रॉब्लम.
सर,
ठीक है,
आपका कांटेक्ट नंबर मिलेगा क्या?
मैंने चैट बॉक्स में डाला
Sir has
हुआ है.
already shared in chat box.
सर नहीं दिख रहा सर।
नहीं दिख
I am sharing now.
रहा. सर
I
विल
think
शेयर,
sir
नो
he has
प्रॉब्लम.
joined late. He has joined late, sir.
यस सर,
ओके,
यस सर, यस सर, मैं थोड़ा लेट ज्वाइन हुआ इसीलिए।
कोई बात नहीं. आप अपना ये कर दीजिएगा, आप अपना डिटेल दे दीजिएगा उसमें, व्हाट्सएप पे भी तो मैं आपको भेज दूंगा जो भी मेरा.
मेरा
अगर अभी
नंबर
आपको,
मैसेज बॉक्स में लिख दूं क्या सर?
हाँ, लिख दीजिए प्लीज.
सर आपने बताया कि सर और एक क्वेश्चन था
हाँ.
कि नैनो पार्टिकल्स की एंटीबैक्टीरियल हम एक्टिविटी देख सकते हैं, तो वो वाटर में डिसॉल्व नहीं हो सकते फिर?
नहीं, एक्चुअली आप मेटल्स नेवर डिसॉल्व इन वाटर.
यस सर, देन यू कैन यूज सी कि हम DMSO यूज कर सकते हैं,
वी
लेकिन
आर यूजिंग DMSO एंड आल्सो सोनिकेशन. वेरी इंपॉर्टेंट इज सोनिकेटर यूज करना पड़ता है वो, बिकॉज इफ द पार्टिकल विल नॉट डिस्पर्स, हाउ इट विल बी यूज्ड, आई डोंट नो, वेरी डिफिकल्ट.
ओके
और हमने
Yes,
बिना उसके
sir.
कभी
One
कभी
question is
किया नहीं.
related to sonication, sir. From the Tasneem Bohra. What is the significance of sonication in green synthesis of nanoparticles? Hello. Alok, sir. One question related to the sonication. What is the
ये
significance
बंटू
of
सुनील आउट.
sonication in green synthesis of nanoparticle?
थोड़ा बाहर उसे बोलिए, क्या सर क्या बोल रहे थे एक्चुअली, मैं थोड़ा.
One question is here. What is the significance of sonication in green synthesis of nanoparticle?
हेलो.
Yes, Priyank, you may ask question. Just unmute yourself,
Yes,
Priyank.
sir. Good evening. Very good evening to all the delegates. Sir, myself Priyank Kanodia, and I am a PhD research scholar from Veer Narmad South Gujarat University, Surat. Sir, I am working on the probiotics microorganisms, and on this
ओके.
probiotics microorganism, the mainly work is focused on the antimicrobial peptide, which is generally produced by this probiotic microorganism.
यस
Sir,
यस यस.
and objective is that to produce these antimicrobial peptides from this probiotics microorganism and to check the effect activity against the different type of pathogen. So, sir,
या
I
यू
had
कैन डू.
checked the antimicrobial activity of these antimicrobial peptides again pathogen, all the pathogens of the lab.
ओके
But sir,
ओके
my second objective is to check the this antimicrobial activity against multiple drug resistant organisms. Sir,
ओके
so
ओके.
my question is to that, sir, whether which from which source I have to collect the pathogens to check the activity of this antimicrobial
You
activity. It means
you see, you have to. Yeah.
because the
Please.
probiotic microorganisms are diverse in our whole body. So मेरा ये क्वेश्चन है कि मैं कौन से सोर्स का माइक्रो पैथोजन लूं कि ताकि मेरा एंटी माइक्रोबियल पेप्टाइड है वो चेक कर हो सके मल्टीपल ड्रग ऑर्गेनिज्म के।
तो देखिए, मल्टीपल ड्रग रेजिस्टेंस है, EMR जो बोलते हैं हम जिसको.
हाँ, यस एग्जैक्टली।
Basically, these bacterias are found in those patients who are taking some drugs. Basically, some antibiotics or some you can say some infectious patient can basically they take this type of drugs.
सर
So you can collect.
बेसिकली
I have collected a
या
Klebsiella sample from pus of a secondary infection from my relative, and it was a multi-drug resistance bacteria, and no drug is working on that. So you can contact somebody, or you can take help of some medical
What?
personalities for this work.
Sir, I can take the multiple drug resistant organisms from UTI pathogens or not?
Yeah, yeah, yeah. You can take from
But sir,
anywhere.
the main point is that I had isolated these peptides, antimicrobial peptides from probiotics. So the question is that these probiotics are diverse in our whole body. So from which organs मैं ले सकता हूँ कि जो उसमें से मल्टीपल ड्रग रेजिस्टेंस मिल सके, क्योंकि प्रोबायोटिक्स होल बॉडी में डाइवर्स
तो
होते हैं
सबसे
ना.
सबसे बेस्ट तो इंटेस्टाइन से लीजिए ना आप, जो भी बैक्टीरिया रहते हैं
हाँ.
मोस्टली प्रेजेंट इन इंटेस्टाइन, दैट विल बी बेटर फॉर यू.
हाँ, because this can be the
हाँ,
question of
क्योंकि
defending
प्रोबायोटिक्स
in my
दे
Bible.
बेसिकली वर्क इन योर गट्स. तो आप गट्स के फ्लोरा के किसी
What?
भी बैक्टीरिया को टारगेट कर सकते हैं ना.
Guts flora के MDR.
गट फ्लोरा कैन यू कैन आल्सो टारगेट, नो प्रॉब्लम.
हाँ, मतलब जो गट फ्लो में जो माइक्रो ऑर्गेनिज्म होते हैं वो एमडीआर हो चुके हैं, उनके सामने मेरा पेप्टाइड का एफिकेसी चेक कर सकता हूँ.
हाँ हाँ,
तो ये मुझे सैंपल मिल जाएंगे ना सिविल हॉस्पिटल है
ये
और ऐसा
हमको तो
कोई.
मिल जाते हैं. वी हैव सम कॉन्टेक्ट्स विद गवर्नमेंट मेडिकल कॉलेज, और द पेशेंट्स व्हिच वर कमिंग फॉर द ट्रीटमेंट, इनको बहुत मल्टीपल इन्फेक्शंस है. Those
ओके.
patient have AMR or this type of bacteria. You can use CLSI standard for that also.
Yes,
You
I am
can
using that CLSS standard for
you
checking of this. But sir,
you
can
can
I
use that.
contact you for this if I cannot get the sample of this MDR from sources, then?
Okay, I will give Klebsiella, no problem. Klebsiella तो मेरे पास ready रहता है एकदम. It was my
Okay.
favorite bacteria.
Okay, sir.
हाँ.
And sir, I am synthesizing the multiple interventions of this.
Hmm.
So one of the objectives of my PhD is to make the nanoparticles of this probiotics pellet. Basically, what
Oh,
I am is doing is that I am centrifuging the probiotic microorganism and taking the cell-free supernatant as an antimicrobial peptide. But the remaining
okay.
part, that what is the pellet? So I am thinking that there is a situation or anything that I can make nanobots or any nanoparticles from this pellet.
Yeah, you can make it, no problem. But
But
for synthesis of any nanoparticle, you have to use some liquid formulation, because because solid material cannot reduce anything. Because when जब ये liquid form में रहेगा तभी अच्छे से काम करेगा. That pellets cannot be used. You have to dissolve the pellet, and again you have to check for the antimicrobial activity.
so what I can do is that I have to dissolve these pellets with the help of this PBS postal buffer saline.
Yes, yes, you can dissolve it in some buffers, or anything which is very water soluble, or you can use also organic, or organic solvents also. है
So
ना?
my objective is to make is that to entrap this antimicrobial peptide in nanoparticle,
Yeah.
and this nanoparticle would
And
be acting
have
as
you
an.
seen? Okay, have you seen the stability of this peptides? Because the peptides, if it is a protein source, or what type of constituent it is, have you checked it or not?
No, no, sir. My next question was this also, that I have to check this antimicrobial peptides stability. So LCMS
Yes,
will
you
be
have to.
good.
If it is a protein, it's very difficult to stabilize it, because protein cannot be stored for long time. आपको
Yes,
lyophilize
sir.
करके
Exactly,
रखना पड़ेगा. You have to lyophilize. हाँ,
sir.
you have to
तो इसका
lyophilize that protein,
हाँ हाँ. Basically, these
or whenever
are
you want, you
the
can.
हाँ.
Yeah.
Basically these antimicrobial peptides are only the proteins, bacteriocins.
Okay. Then you have to
So
go for, first of all, you have to go for lyophilization process, where you can lyophilize these proteins.
hmm,
Or whenever you want to synthesize some nanoparticle or anything, then you have to dissolve it, and you have to put the salt which you want to reduce. That's
okay,
all.
sir. Okay,
Mhm.
and sir, whether to know that which protein is present, I can check. I can go for the LCMS for these antimicrobial peptides.
Yes, you can check it for, or you can go for SDS page also. Or now you can use the new techniques called HPTLC.
HPDLs. Okay.
HPTLC you can use.
Okay, sir.
HPTLC will give you the molecular weight of that protein, or you can decode those protein and find out what type of peptide it is.
Yes, I had already sequenced my organism. Some of the I have two organism. Basically, one Lactobacillus plantarum, and second is that is Hendrixia coagulans.
Hmm, 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 hmm. No,
So
that
these
is
two
the very
organism is
no
showing
you.
maximum
uh,
antimicrobial activity against those pathogens.
okay, okay.
So I'm now checking. My next step is to check this how much amount of protein content and what are the composition of these antimicrobial peptides. Okay. I
Yes.
for the protein content I had run Bradford assay and it is
You can
showing the
find out the
significant
total
value.
protein. You can find out the total protein
Yes.
by any Laurel method or Bedford method, or you can also see what run on HPTLC will help you to find to isolate those peptides which has with standard, or if you have don't if you don't know what type of peptide it is, then you have to sequence it. Otherwise, if you know
Okay.
the molecular weight of the peptide, you can run it in HPTLC also with standard.
Yeah. Thank you, sir.
Thank you. Hello.
Hello, sir.
Yes, sir.
Mhm. Yes, madam.
Hello, everyone. So, I'm currently working on green synthesis of nanoparticles with selenium and magnesium. But the problem is we are not getting exact size of nanoparticles even after going for zeta potential testing. And we are also not getting a single bell-shaped peak efficiently for any of the metals. So, any suggestion, anything
You are
what we
not
can do
getting
about this?
the nanoparticles in that range, basically less than 100 nanometer, madam.
Yeah, yeah.
So, which material you are using for synthesis?
Uh, For synthesis, we are doing the green synthesis. So we
No,
are
green means
using
what?
for
The plant material or any bacteria? Or,
ficus. Yeah, yeah, we are using the ficus bengalensis, the aerial root of it.
it's a plant.
Yeah, yeah, it's a plant.
So, you are how you are processing, madam? You are drying those leaves, or means what are the basic steps you are doing? I don't know, but we are basically drying those leaves and making powder, and after that we dissolve it into some solvent, or and we are using it, or we give dissolve in water for reduction of these
Uh,
nanoparticles.
yes, sir. So basically, what we did was we took the aerial roots of the ficus bengalensis plant and
Okay.
we chopped it and we grinded it with the help of mortar pestle and we
Mm-hmm.
dissolved it in distilled. So we got the water aqueous extract and then we
Okay.
proceeded for green synthesis of it.
Okay. Okay. So how much time you are keeping it for the reaction?
So for selenium, we kept it for 1 hour at 120 RPM, and for magnesium nitrate, we used magnesium nitrate as the salt precursor, and we kept it for 3 to 4 hours at 70 degrees Celsius.
No, the time is very less, madam. Actually, now we have synthesized selenium by actinomycetes, and we
Okay.
have kept this reaction for more than 24 hours. Or you can say, sometime we are keeping this cyanobacteria and this salt for more than 6 to 7 days also, because some extra. I mean, it means the enzymes, or you can say the metabolites which are in the water, it will take time to reduce it. So two,
Okay.
3 hours is very less, ma'am. I think so. You can
Okay. And
increase your time.
RPM, is it okay, or we should increase the RPM?
RPM means for what you are using RPM?
For stirring,
That
we are
can be done on
using for
magnetic stirrer also, madam. No problem. A simple magnetic stirrer is also sufficient for that.
okay.
Yeah. You have to see the color change, and if the color change take time, you have to wait for it, ma'am. That's important.
Yeah, because the color of our extract is like brick brown in color, and
Okay.
after I have read many research papers, I got to know that selenium also shows a reddish color
Oh,
after
that's
the synthesis. So
mm.
it is very difficult for me to check the color change.
Okay, 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 okay. That means the reaction time is very less, so that you are not getting this
Yeah.
particle, because we are getting more than less than 10 nanometers. Now this we are getting 5 to 4 nanometer size. But with the
Yeah.
biological reaction time is greater than chemical reaction, because biological material take time to adjust itself. If it is a living thing, then it will interact slowly with the material. Mhm.
So now come. Yes, there is any question?
Good evening, sir.
Good evening.
This is Shraddha Dehure from VV University. I'm PhD research scholar. Sir, I wanted to know whether traditional zinc liquid biofertilizer is better or zinc oxide nanoparticle will be beneficial for crops. Anyone can guide me?
Excuse me, what do you want to know?
सर मुझे ये
Hello.
पूछना है कि जैसे जो जिंक ऑक्साइड नैनो पार्टिकल की अगर हम एप्लीकेबिलिटी क्रॉप्स पर लेते हैं तो
हाँ
क्या वो ट्रेडिशनल बायोफर्टिलाइजर से बेटर एक्ट कर सकते हैं या हमें दोनों को मिलाकर से कुछ टेक्नोलॉजी डेवलप करनी चाहिए? क्या इस पर आप कुछ गाइड कर सकते हैं?
आपको क्या लगता है, बताइए पहले. What do you think?
सर एक्चुअली मेरा जिंक लिक्विड बायोफर्टिलाइजर पर पीएचडी
नहीं
है
तो मैं
और
आपको
मैं
एक आइडिया
कुछ
दूं क्या? मैं आपको एक आइडिया
जी
दूं
सर
क्या?
जी सर
देखिए, करेगा, होगा, ये कैसे पता चलेगा? साइंटिफिकली वी हैव टू प्रूव इट कि
या
होना चाहिए. देन व्हाट वी
हाँ
बेसिकली आई हैव वर्क फॉर पीजीपीआर और इंटरेक्शन ऑफ प्लांट एंड माइक्रोब्स फॉर मोर देन 10 years. तो मैंने एक चीज देखी
यस
है
सर हाँ
कि साइंस में बहुत सारे कॉम्बिनेशन है. ये जिंक डालो, ये डालो, ये थोड़ा सा वर्मी कंपोज मिला लो. बट
हाँ
what are the impact of this nanoparticles or what are the impact of this biofertilizer on the plant depends on the plant's health. जैसे मैंने आपको खाना दिया
हम्म
और मैंने वही खाना खाया और मेरा वजन 90 किलो हो गया और आपका वजन 85 किलो रह गया. तो
जी सर
इफेक्ट किसके ऊपर आ रहा है? अगर कंसिडर करेंगे तो मुझ पे आ रहा है. मीन्स आई एम रिस्पॉन्डिंग बेटर देन यू.
हम्म
तो समझ रहे हैं? आपको प्लांट से पूछना पड़ेगा कि भाई तुझे क्या अच्छा लग रहा है. तो यू कैन गो फॉर द रूट लेंथ, शूट लेंथ एंड क्लोरोफिल कंटेंट एंड हेल्थ ऑफ द प्लांट बाय मेकिंग सम कॉम्बिनेशंस. कौन सा बेटर रहेगा, यू विल डिसाइड इट. और प्लांट विल टेल
ओके
यू कि ये मेरे लिए अच्छा है.
ओके ओके सर
हाँ, मीन्स अभी मैं नहीं बोलूंगा. आप समझ गए कि नहीं अब मैं क्या बोलना चाहता
जी,
हूँ?
जी। सर आपने
हाँ.
मुझे पहले भी गाइड किया है सर, दिस इज़ श्रद्धा रडके, आरजीबीसी में हम
अच्छा.
साथ में थे।
हाँ
सर
हाँ, श्रद्धा. अरे क्या कैसे बेटा?
ओके सर
आफ्टर
थैंक यू
वेरी
सो
लॉन्ग
मच
टाइम.
सर। आई विल यस सर, आई विल go for pot experiments now and I will
यस, यू
continue.
कैन फाइंड आउट वेदर द प्लांट इज रिस्पॉन्डिंग और नॉट. मैं तो ठीक है, मैं खाना दे रहा हूँ, वो खाना
जी,
खा रहा है, लेकिन उसका फायदा हो रहा है कि नहीं, ये तो प्लांट ही बताएगा बेटा कॉम्बिनेशन के बाद, है ना?
जी, जी सर। थैंक यू
ओके
सर।
बेटा, ओके बेटा, बेस्ट ऑफ लक.
So now we come to the end of the discussion. So any question put in the chat box. So now I request.
Yes, sir. I have a question.
Yeah, Tapas.
Okay, sir. Good evening. My name is Tapas Kumar Das. I'm actually an MSc student. Biotech. I'm doing a course in biotechnology. My question to you is this: are there any professor or scientists conducting field research has in bioprospecting of extremophiles in where they collect samples from the diverse ecosystem where extremophiles are grown and then take the sample to the lab and then sequence the DNA and find proteins which can be used in therapeutics and other industrial applications. I just want field experience as an internship,
From which
if it is possible.
institute you are?
I am from
Tapas
Maharaja
Kumdas from.
Surajwal Bridge. Yes, sir. I am from Maharaja Surajwal Bridge University, Bharatpur, Rajasthan.
Okay.
Yes,
So in Kolkata.
in Satyabhama University, Chennai, he actually does the bioprospectum of marine microorganism, actinomycetes, halophilic bacteria.
Thank you, sir. Can you write this on the comment section? I want to note it.
And
Excuse me, can I say something about this? And
yes, sir. Please.
what do you want to know about extremophiles? Because extremophiles have lots of diversity in metabolites also. So when I was working on extremophiles, we have
Yes, sir.
isolated some proteins. Now I am also working on uh, a bacteria which can precipitate calcium, and we are going for some biomaterials which is used in crack healing of this cement. Now paper is coming in that field, and we have found out that this whenever the crack was there in the building, the bacteria grows and it will seal those crack by forming calcium oxalate or calcium chloride.
Yes, sir.
So, what metabolites you are targeting, that is very important.
Sir, I am
Because
actually, yes, sir.
Mhm.
Sir, I
because
am
one of my
actually,
friend, he has targeted a protein which is
yes,
very
sir.
nice protein, and he has make a patent, and that protein can be used in paint industry, which enhances the surface area of the paint and strengthen the cement also. So he has patented it, and the company has taken it for more than crore rupees. So it has a lots of potential. Extremophiles can do anything, but
Yes,
what
sir.
do you want? That is very important.
Sir, I don't want anything in specific. I just want field experience, sir, to know and get experience and have academic connection to those scientists who actually conduct field research. I don't have any specific interest in microbiology. I want to do research in diverse and big field. I don't have any specific requirements. I just want academic connections from this. I think
Yes,
extremophiles has great potential. That's why.
yes, extremophiles have great potential. I have worked for more than 2 years in extremophiles condition in NBI, and we have isolated most of the bacteria from salt-loving bacteria or even Manikaran hot spring water. आपको पता होगा हिमाचल में है जो.
Yes, sir. Yes, sir.
हम्म.
Hot springs.
So you can contact some scientists, like people who are working on extremophiles, and it is
Yes, sir.
sometimes very difficult to isolate it because of the variations in the temperature and the conditions.
Yes, sir.
So
So, can you recommend me to those scientists, or you share me your email? I will contact you.
yes, I will give some numbers. You can contact those people who are working on
And
that.
also in Kolkata, we may contact Dr. Ridhiman Ghosh and Dr. Masurre Alam. Both are working on extremophiles in Kolkata,
Yes, sir.
West Bengal. So now I would like to request our Dr. Prakash Salami sir to conclude this session. Sir, hello.
Yeah, in fact, it was a wonderful session. We could see today very good participation. Also, almost
Today more than 50 participants joined.
exactly. So this itself shows the importance of this subject, nanotechnology. Though nanotechnology is a multidisciplinary subject, starting with the chemistry till the engineering. But then microbiology has a lot to play. Finally, its application comes to the microbiology. Nanotechnology again starts with the microbiology, I would say. Nanoparticle synthesis or green synthesis of nanoparticle, you know. And one of this participant rightly mentioned that how you look for the application. And that is what actually the nanotechnology has a emerging scope in a days to come. The MBS, what it has taken the lead in this roundtable discussion. I'm very happy that Tiwari ji with a very smiling face is conducting these activities. And
Thank you so much, sir.
he is helping the several actually research scholars how they can actually come forward and then clear their ideas. Maybe they may not be able to get this clear idea from their guide, perhaps.
Yes, sir.
But then the MBS also act as a co-guide for them. We are there as a resource for them. We are here to help them out, through this MBSI platform. Only my request is better we limit always within the topic of the discussion. Otherwise,
Yes, sir.
it's never ending, But of course, those who has
Today's topic was only on nanotechnology.
exactly. Of
So,
course, within the purview of microbiology, whatever they are raising the questions and discussing, most welcome. And it was a very wonderful session, I would say, today. And I
thank you so much.
look forward to the similar session in the days to come. Every Tuesday, 7:30 p.m. Thank you.
Yes, sir. And yes, Saurabh Chaudhary, thank you so much, sir. You may get the recorded session in our YouTube channel, Microbiology Society YouTube channel. You may get this recorded session because this one is the live streaming, so you may get the recorded session there. And Priyank, sir, we need more lectures on this topic.
Yeah, possible. I think we can modify a little bit more.
So I requested Dr. Alok Rai sir and Ganga madam to join the next session also. Is it possible from you,
Yes, sir.
sir?
Please, it was very truly worthy lecture and presentation. So one of my the question was solved today. So thank you,
Yes.
Alok sir and Bripesh Tiwari sir and MBSI Society. Recently, I got scholarship from this Microbiology Society India
Okay.
For what purpose?
Congratulations also.
for PhD.
Oh, wonderful.
Yeah, thank you, sir.
Thank you, friend.
Sorry for disturbing. Sir, I want to actually ask, can you give me the name of the scientists who are conducting field research on extremophiles? So I
Actually,
didn't listen it
I will share
previously.
my one of my friend working in Scotland. He is working on sea diversity
Yes,
and
sir.
different depths of sample he is collecting from seawater. So you
Yes,
can
sir.
contact him. I will give number, no problem. There are lots of people working
So,
on this type of work.
Thank you,
Yeah.
sir. Thank you very much, sir.
and also, Tapan, I have shared my WhatsApp number. Okay, just say one hi, then I will share the name of the scientist from the Kolkata.
Yes, sir.
Okay.
Thank you, sir.
So, Alok, sir, is it possible from your side to join the next session, means next Tuesday, so that you can continue with this topic?
Yes, sir. Okay, okay. I will try my best, sir. I will try.
Sure, sure.
This is first time I have joined. I am very happy to
Yes.
discuss with all of you.
So then you can continue with this topic, sir.
Yes, yes.
Next session, because there are questions of the participants that
Yes,
we
yes,
can
we
take in the
can
next
go
session.
more detail.
Yes, sir.
With reference to the experimental part, how this
Sure.
was done, and then what was the results, what the controls were kept, how the biological activity efficiency was tested.
Yes, yes. I can share some of the my papers data, or you can see. I can.
So in the next,
You can explain the
Dr. Alami, sir.
tell me.
In the next means week, we can start with the presentation of Dr. Alok Rai, sir.
Yeah, we
And
can
then
do that.
Yeah.
10 to 15
First, we
minutes
can.
more.
Yeah.
Compact one, and then we can have a question and answer on that one
Sure,
also.
sure, sir. That will
That
help
also
the
can be
researchers
useful
more.
information. As I already told that everything starts with the microbiology, so nanotechnology is not an exception. In other words. Exactly.
So thank you, everyone.
So those who are working in the similar line, they can also be informed by their friends, those who are here today. So they will have a better audience group.
Sure. So Dr. Alok, sir, I will share the link for the next week with you personally. Hello. Am I audible to you, sir?
Yes, sir. Yes, sir.
Okay. So we'll start in the next week. We'll start with the presentation, sir, and then we'll go for the discussion part.
Okay, sir. I will
Thank
share
you.
some of the data which we are working on, and so
Yes,
that
yes.
we can help those students, those who want to do it, sir. No problem.
Sure, sir.
Thank you, sir.
So thank you all the participants for joining today's session. My special thanks to Dr. Alok Rai, sir, Dr. Ganga madam, and all the participants, means respected professor or scientists who have contributed for the today's session. And also my special thanks to Dr. Prakash Alami, sir.
Thank you.
He is the pillar of this roundtable discussion, who participated every week roundtable discussion actively. So thank you everyone for joining today's session. So next week we'll continue with this topic and Alok sir with the Alok sir presentation. Thank you so much.
Thank you, sir.
Thank you, sir.
Thank you. Thank you, everyone.
Thank you, sir.